అమ్మా సుజాత, పూజ.
Madam, good morning.
Good morning. Ma'am, good morning, ma'am.
Madam, వినిపిస్తుందా మేడం?
వచ్చారా అమ్మా మేడము? మల్లిక గారు వచ్చారా?
వచ్చారు.
సుజాత, నువ్వు అన్మ్యూట్ చేసుకోవాలి. వినబడట్లేదురా? ఏం వినబడట్లా?
మేడం పూజ మాట్లాడడం వినిపిస్తుందా? పూజ మీరు ట్రై చేయండి.
మేడం నేను మాట్లాడుతుంది వినిపిస్తుందా?
గుడ్ మార్నింగ్ మల్లిక మేడం.
గుడ్ మార్నింగ్ మ్యామ్.
నాకు వినిపిస్తుంది పూజ. బహుశా మేడం కి ఆడియో ఏదో ప్రాబ్లం ఉన్నట్టుంది. నేను ఒకసారి
రాలేదు మేడం.
నా వాయిస్ రావట్లేదా?
అమ్మా సుజాత
మేడం మీ వాయిస్ మాకు రావట్లేదు. శ్రీదేవి మేడం.
సుజాత నేను అన్మ్యూట్ కూడా చేశాను. నాకు ఇక్కడ కనిపిస్తుంది. ఇప్పుడు వినబడుతుందా?
వినిపిస్తుందా మేడం?
నువ్వు మాట్లాడేది ఏం వినబడట్లా. సార్ ట్రై చేయండి ఒక్కసారి. మీరు మాట్లాడేది నాకు ఏం వినబడట్లే. సంథింగ్ అంటే సెట్టింగ్స్ ఏమన్నా వాల్యూమ్ తగ్గించాలా?
పెంచండి మేడం, వాల్యూమ్ పెంచండి.
ఓకే, ఇక్కడ ఏదో రెడ్ లైట్ వెలిగింది. ఇప్పుడు ఓకే, ఇప్పుడు మాట్లాడరమ్మా.
మేడం వినిపిస్తుందా మేడం?
వినబడుతుంది రమ్మా, వినబడుతుంది.
ఓకే మేడం.
గుడ్ మార్నింగ్ మల్లిక గారు.
గుడ్ మార్నింగ్ మ్యామ్.
గుడ్ మార్నింగ్. Sujata, shall we wait for another 2 minutes?
Yes madam, we will wait for 2 minutes, then we will start.
Okay.
Good morning sir.
Good morning madam.
Babu sir, namaste.
Namaste
sir.
How are you madam? How is going on?
Fine sir, fine. We are fine sir.
Good morning Dr. Sridhar madam.
Good morning sir. How are you?
Yeah, fine ma'am.
చాలా రోజుల తర్వాత చూస్తున్నాం సార్ మిమ్మల్ని.
ఆ టైం లేదు మేడం. సో రొటీన్ వర్క్స్, యాక్టివిటీ బిగిన్ అయింది కాబట్టి ఇంకా దాంట్లో బిజీ అంతే.
అవును. ఓకే ఓకే.
కాలేజ్
స్టార్ట్ అయిపోయిందా సార్? ఆన్లైన్ క్లాసెస్ ఏనా లేదు అంటే మామూలు క్లాసెసా?
మామూలుగా క్లాసెస్ బిగిన్ అయ్యాయి కదా మేడం.
ఆఫ్లైన్ వచ్చేస్తున్నారా పిల్లలు?
అవును. సో 90% అటెండ్ అవుతున్నారు.
ఓకే ఓకే ఓకే.
సో ఎగ్జామ్స్ కూడా జరుగుతున్నాయి.
ఓ సరే, మాకు డిసెంబర్ లో స్టార్ట్ అవుతాయి సార్. మా స్టూడెంట్స్ యూనివర్సిటీలో స్టార్ట్ అయ్యాయి. బట్ ఫార్మసీ వాళ్ళకి ఆన్లైనే, డిసెంబర్ లో ఆఫ్లైన్ స్టార్ట్ అవుతాయి.
మీరు మన ఇది గవర్నమెంట్ వీళ్ళు ఇన్సిస్ట్ చేస్తుంది, యూనివర్సిటీ వాళ్ళు కూడా ఫోర్స్ చేస్తా ఉన్నారు. అలానే ఇంకా ఎగ్జామ్స్ పెట్టారు, ఇంకా వాళ్ళు ఎలా ఇంటికి పోరు కాబట్టి మనం కూడా కంప్లీట్ చేస్తున్నాం మేడం క్లాస్.
ఓకే ఓకే ఓకే.
ఓకే మేడం, గుడ్ లక్. ఐ థింక్
గుడ్ లక్. థ్యాంక్ యూ సార్. థ్యాంక్ యూ.
నమస్తే
సార్. నమస్తే
మేడం. స్టార్ట్ చేసుకుందామా?
చేసేద్దాం రామ్మా. జాయిన్ అయిపోతూ ఉంటారు వాళ్ళే.
ఓకే ఓకే మేడం.
ఓకే.
Pleasant good morning dear participants. We welcome you to the day 5th of the 1-week FDP program conducted by Institute of Pharmaceutical Technology. Today it's my pleasure and I am really happy to invite Dr. A. Mallika as a resource person for the morning sessions. On behalf of Institute, on my own behalf, I welcome Dr. A. Mallika and thank you for accepting to act as resource persons in this faculty development program. Mallika has lots of experience in computer-aided drug designing. Before joining NIPER Hyderabad, where she served for 7 years, she worked as research associate at Dr. Reddy's Institute of Life Sciences. Recently, she has started her own training center, that is Mars Training Academy. She is a founder and instructor in Mars Training Academy. She has completed her PhD under the guidance of Professor Sriram in BITS Hyderabad campus. She also guided 3 PhD students and 26 master students for thesis. She has an excellent record of publications in peer-reviewed journals. More than 55 publications are credited to her account. She is elected as Associate Fellow of Telangana Academy of Science in 2017. She also received Best Research Scholar for the year 2012 from Pharmacy Department, BITS Pilani, Hyderabad campus. This was sponsored by the industry Bharat Biotech. She is a motivated and energetic computational research with over 10 years of experience in this computer-aided drug designing program. Her research interests are mainly cancer, tuberculosis, and obesity. Now, currently, she is focusing on employing machine learning models in design process. She is an expert in CAD, Python, and machine learning, and she is particularly committed to train for people to utilize these applications in field of pharmacy. Thank you once again, Mallika. We welcome you to this session. I will hand over this session.
Thank you for the introduction. I thank all the organizers for giving me this opportunity to have a presentation. Can we start now?
Yes, please.
So to maintain the bandwidth, I'm just muting my video. Good morning, one and all. I think from past 4 days, people are giving a lot of explanations and the lectures and machine learning and informatics in drug discovery. Today, I'm going to give brief view about role of informatics and machine learning in drug discovery process. Before going to start the session, I'd like to start my session with a great quote given by the Nobel laureates in 1998, stating that in the real world, this could eventually mean that most chemical experiments are conducted inside the silicon of chips instead of the glassware of laboratories. Turn off that Bunsen burner, it will not be wanted in 10 years. So now we can imagine that we are 20 years away from 1998, and now most of the experiments are going on in silico. It's not that we are not doing wet lab experiments, but the content and the amount of wet lab experiments we are doing has been tremendously reduced, and most of the experiments we are conducting on in silico. So let me give brief about the progress in the drug discovery process. As all of you know, before 18th century, there was a Chinese legend, and the drug discovery was stated by that tasting to testing. So the Chinese legend, Shen Nong, used to taste lot of herbs, hundreds of herbs in a single day, and try to give the use of those herbal drugs by tasting itself, whether they are beneficial for the human health or whether they are going to be toxic for the human health. And he used to meet a lot of toxins, and these toxins he used to use for killing the cancer cells, and some of them will be useful for the beneficial of the human being. That was the drug discovery process before 18th century, that can be stated as a tasting to testing. In traditional drug discovery process after 18th century, we mainly depended on the plant resources and the microbial resources, and we used to isolate the plants or itself the total plant extracts and try to find out what is the benefit of that particular plant. We used to screen on various methods like various pharmacological activities like cancer activity and anti-diabetic activity, and try to find out which type of activity it was showing. Based on that, we used to claim that that particular plant is going to be used as either antibacterial or anti-diabetic or anti-malarial. And the method was like purely based upon the screening process. It was very time consuming and expensive because we have to screen the plant extracts, and the isolation of molecules are also very not so easy. Hence, it will be a very expensive process and the success rate was very low. After traditional drug discovery process, because of growth in the informatics and the computational tools, we used lot of computer aided drug design processes that can be also called as a rational drug design process, because now the scientist has got an idea how the protein looks and what is the sequence and what is the active site. So based upon the idea of the active sites, he used to design the molecules which will specifically bind to the particular protein. Hence, that will be called as a reason-based drug discovery process, where if you use computer tools or the softwares, then it will be called as a computer-aided drug design process. And this will be target specific, as we are using the structure of the protein structure as an initial point to design the molecules. It can also be called as a structure-based drug discovery process. Rational drug discovery and the computer-aided drug discovery process is very fast and automatic, and the cost required to design the molecules by using this process is very low and the success rate is very high. Now we are in the era of the artificial intelligence and machine learning, where the computers used to take lot of decisions. It's not that computers itself will take a decisions, but we are going to train the computers in such a way that they can understand the problem, and whenever we give new problem to the computers, they can give some conclusions and answers to us from where we can design the molecules. So this has become now automatic process of drug discovery, not only in drug discovery, in various fields the artificial intelligence is implementing. The main important feature required for doing the artificial intelligence or machine learning programs is data. This data, we are going to see which type of data we are going to implement for the machine learning models. And this process, compared to the CAD process, it is going to be very fast and automatic, but it requires little bit of programming language experience. So this is how the progress has been happened in the drug discovery process, starting from tasting to testing, and now the era of the computers, where the computers are going to take a lot of decisions and they can help us in designing the molecules. Let me give a brief idea about how the informatics has played a major role in the drug discovery process. So we know that for any discovery process, first of all, we have to identify the disease condition, on which disease we want to work it out. Then we have to isolate the protein which are involving in the disease condition and which are important to cause the disease. So this can be done by the biologist or the pharmacologist, who can take out the proteins and isolate the proteins. They try to understand the function of that proteins in that particular pathway and analyze how much it is going to be impact for the disease progression. It almost takes 2 to 5 years. During this case, the crystallographer used to understand the three-dimensional structures of the proteins and get the X-ray structures to understand what are the binding sites are available. After finding the protein, once we decide this particular protein is going to be involving in the disease condition, we try to find out the drugs which are going to be effective against disease protein. So these drugs may be inhibitors or the activators, or simply the substrates which are going to modulate the protein function so that you can alter the disease condition. This almost take 2 to 5 years. Here the medicinal chemist or organic chemist will play a major role to understand which type of molecules are going to be binding to the protein structure and how to design the molecules, also synthesis. So sometimes the synthesis may be possible, or sometimes we will stuck up at certain points, but still we will try to find out the molecules which are going to alter the protein function. Once we find out a hit, this can be like a lead, hit, and drug molecules. So from lead we will go to hit to the drug molecule, And these molecules we will test in a preclinical testing in animal models. Most of you may be knowing there are 4 varieties of clinical trials are going to be happen. So this takes around 1 to 3 years. Once the preclinical testing is over, the formulation optimization can be done by the Sutix people or a formulation scientist in order to bring into a form which can be taken by the human beings. So this is the discovery process. Let's see how the informatics has played a role in each stage of the discovery process. So in case of identification of disease conditions, earlier we used to find out like pandemic conditions used to come. Depending upon that, we try to identify the disease conditions. But now the WHO is maintaining the disease statistics. Based upon the disease statistics, we can identify the disease on which we want to work it out. Sometimes the population will be very low, but that is very important to treat that disease condition, like rare diseases. So you can work it on either pandemic diseases or maybe the diseases which are going to be rare diseases. Once we identify the diseases, based upon the disease statistics of WHO, the next task will be identification of potential targets. So instead of isolating all the proteins and finding out the pockets and the three-dimensional structures, we have certain tools which can help us to find out the druggability of the protein. So here the druggability means how much the protein can be targetable. So for example, in cancer, you have a lot of proteins are available. So among that proteins, which protein is more important for the disease condition? So you can find out the druggability score, which tells about what is the capability of that particular protein to be targeted, either complete protein or the active sites. So if the active sites are very huge number in the protein structure, we can segregate the active sites by finding the druggability of the proteins. So here, instead of isolation, directly you can identify which type of proteins can be targetable and which type of active site can be targeted by using the druggability scores. Then during this process, if we don't know the structure of the proteins also, we have a lot of methodologies are available where we can utilize homology modeling technique and identify the or generate three-dimensional structure, and that three-dimensional structure can be validated by Ramachandran plot and many other parameters. Once you identify that protein structure, then we are going to understand the active site. So that is talking about the druggability at the second stage of the drug discovery process. The next stage will be designing of the small molecule inhibitor or activators. Instead of randomly synthesizing the molecules, we try to understand the active sites and what is the hydrophobicity, hydrophilicity of that protein pocket, and what are the important amino acids which are present in the protein pocket. Based upon that, we try to design a molecule, and this molecule should have the property of drug likeliness. So here the drug likeliness is nothing but the compounds or the molecules which are following the Pfizer rule of 5, like molecular weight and log P, donors and acceptors, everything should be in the range of 5, should be considered as a drug-like molecule. So the drug likeliness score will be calculated for the small molecules, whereas the druggability score will be calculated for the proteins. So once we design a molecules, during this stage itself, instead of directly going to the animal studies, we can predict ADME properties, which are very important before taking a molecule for the preclinical testing as well as for the formulation studies. So this pre-ADME predictions can be done by using various online tools. You have Swiss ADME, pre-ADMET, all these tools are available which can give certain idea about the ADME properties of the molecules. So once ADME properties has been calculated, then we can segregate our molecules for the synthesis process. Based upon the synthesis protocols available and easy to synthesis, we can start synthesizing the molecules. Instead of synthesizing 1000 molecules, we can shortlist and we can synthesize limited number of molecules which can be having drug likeliness properties and acceptable ADME properties. These molecules will be further taken for the formulation optimization. So earlier we used to do lot of formulation optimizations, taking the small molecule and the polymer, different proportions and different solvents. But now the informatics tools are available where you can understand interaction between the polymer and your drug molecule by in silico. You can find out whether these two molecules, your drug molecule and polymer, can have a physical interaction or a chemical interaction, how strong is that interaction, whether that interaction will be reversible or irreversible, as well as the stability. Based upon the dynamic studies what we perform between the drug and the polymer with the different proportions, we can able to understand which type of polymer is compatible with the drug and what type of interactions it is forming, whether these interactions are strong or not. If they are strong, how far they are strong. So all these calculations can reduce the process of formulation optimization. So with this availability of the informatics, the drug discovery process has been tremendously reduced from 10 to 13 years to the 2 to 3 years. So this is about the discovery informatics. Now coming into the developmental stage. So once the formulation optimization has been happened in the laboratory, the molecule can enter into the human clinical trials, various clinical trial stages. And once the FDA approval happens, the drug comes into the market. From clinical trials to the market stage, even after entering into the market, we generate a lot of data, clinical trials data at 4 stages with a different population, different races, and different combinations. So all this data will be available in the clinicaltrials.government site. So from this site, we are utilizing only limited amount of the data for our conclusions. So with the development of big data management, acquisition and analysis, by using artificial intelligence, machine learning and deep learning models, we could able to analyze the data which we already developed by various clinical trials. So instead of doing again the clinical trials, we can utilize the already existing data and we can get a conclusion and comparison between different clinical trials with different combinations. So the informatics, but the data which we develop in the clinical trials is very huge. So we require computational tools or the machine learning models which can handle big data. It's not that without big data, without machine learning, we cannot do the clinical trials. We can do, but some errors can happen when you compare the dependent and independent parameters. So this artificial intelligence and machine learning tools has reduced the error rate as well as increased the success rate in the comparisons of the data. This is about drug discovery and development process. Now let's see what are the advances happened over here. So in case of testing, earlier we used to depend upon the animal models as well as in vitro enzymatic assays as well as in vivo in vitro cell line studies. So if you want to do such type of screening for many molecules, it is very costly process. But now we have automatic screening technologies where you can have microarrays and body on chip type of technologies. Instead of doing the experiments and killing the animals, we can perform the experiments in body on chip models so that at a time you can get all the information about ADME properties of the molecules as well as how it is functioning inside the particular organ. That is about the testing methodology. Coming to the screening technology, where we have already discussed about the CAD and designing purpose, we use bioinformatics and multivariate analysis models. That is nothing but the databases which are already available can be taken as a starting point and it can be utilized for the drug discovery process. Similarly, we have omics technologies, which we call it as genomic data, proteomic data, and metabolomics data, which is very important to understand the interactions between the proteins because the pathways which are involving in the disease conditions are very complex. If you understand the protein function in one disease condition, it may be upregulated, but in another condition, it may be downregulated. So we have to understand what are the interacting proteins which are making this to happen. So where we have to control that protein expression. So these things can be understood by the omics technologies. These are the advances which are available now in literature, and all these tools are freely available. So before going into the in-depth about the informatics and the artificial intelligence, I just want to brief you why we have to visualize our protein structures in different forms. Because chemists and the drug discovery scientists may know that there are a lot of visualization tools are available in the literature and in the softwares. But why we have to understand and visualize the three-dimensional structure of protein in different forms? If you observe here, there are 4 varieties of representations of the protein structures. The first variety was the simple stick model, where you can understand there are carbons, nitrogens, and oxygens which are having amino acids will be connected. But by seeing this, you can understand what are the how many amino acids are present, and what is their three-dimensional arrangement. But you cannot understand what is the active site and how the size and shape of that active site. Similarly, if you visualize your protein structure in the form of alpha helix and beta sheets, still you can understand how these amino acids has been folded in a three-dimensionally, but you could not able to find out what is the exact pocket of your protein drug molecule. Because if you design a molecule where it is going to be fitting, whether it is going inside the pocket or fixing into the pocket properly, it has association or dissociation capacity or not. All these things cannot be understandable when you visualize in the form of alpha helix and beta sheet. But if you visualize the protein structure by using visualization tools in three-dimensional surface form, you can easily understand that there are certain grooves are going to be present. Grooves are nothing but the pockets. So based upon the pocket, you can understand that whether it is going to be having big size or volume, what are the amino acids, whether these amino acids are going to make any interactions or not. So similarly, the small molecules can also be visualized in the form of 2D and 3D forms, because if you want to understand the three-dimensional pocket, and if you visualize your small molecule in two-dimensional structure, it cannot fit into the it can fit into the pocket, but you have to see whether the volume is exactly matching into the pocket or not. It's like simple lock and key model. When you visualize both in the same format, you can easily understand how tightly they are binding. So this is about the visualization, why it is important to visualize your structures in different formats, so that you will get in-depth information about the active site pocket, size, volume, and the amino acids information. So let's see, if you want to design any new molecule for any disease condition, what are the different methodologies we will follow? First thing is the modification of existing molecules. So where you utilize the information of already existing inhibitors or the activators. You take that as a starting point, try to modify functional groups so that you can imagine that that is going to show a activity. So during this case, modification of existing molecules, we have to take care that whether this particular functional group from hydroxyl to NH2 group we are modifying, whether the protein pocket is having that much volume to accommodate nitrogen or not. So like that, you have to understand the volume of your functional groups and the nature of your functional groups, whether they are hydrogen bond donors or acceptors, electro negative groups. So based upon these considerations, you can modify existing molecules. And second thing was the from natural products, either from plant kingdom or microbial world, marine, animal, or venom and toxins, you can able to isolate or design new molecules. So next method was the considering the natural ligand or the modulator, where you are going to take starting point as a natural modulator. Like if you want to design acetylcholinesterase inhibitors, you are going to take acetylcholine as a starting point and you modify that particular acetylcholine structure so that it can have affinity towards the acetylcholinesterase enzymes. So here you take natural ligands for the receptors and the natural substrates as the starting point. Then another methodology was the existing drugs, that is nothing but me too drug formation. Here we try to enhance the side effects of the molecules. Here we are not going to existing drugs modification and modification of the existing molecules, what we discussed in the first point, are going to be same. But in case of first method, we try to improve the potency of the molecule. But in case of me too drugs, we try to reduce the side effects of the existing molecules. We are not bothered about the potency alteration. So that is called as me too drugs. Next method was the medical folklore method. We used to take traditional claims from the different origins and try to mimic or give the scientific validation for that medical folklores. Because now we have seen when the COVID pandemic has started, people started existing molecules modification as well as taking out the plant products and try to develop a molecules. And some like medical folk force, like traditional claims what we use for the respiratory disease condition, people started giving the scientific validations for the medical folk force. So these are the various strategies we will follow if you want to design new molecules. So in each method, we used to utilize the informatics data. In case of modification of existing molecules, people try to collect the data from the small molecule databases. The small molecule databases include Zinc database, PubChem, ChemEMBL, Drug Bank. There are a lot of databases now emerged. You can find out everywhere, but all are integrated. Natural product information you can collect from the natural product databases and marine databases. And you have natural ligands from the enzyme and substrate databases. Similarly, the medical folk force you can collect from the plant database and medical traditional claim databases. So there are a lot of improvement happened in the informatics since from the 1993. In 1996, DNA sequencing platform has introduced. Then 97 genomics platform has introduced. So if you see like that, improvements has happened in identification of the genomic data, proteomic data. Similarly, on the counterpart cheminformatics system, like collecting the chemical data and segregating and putting it at one place. What is the activity of those molecules and various protein conditions, like PubChem database can give you the information about what are the activities has been performed for individual molecules. So in 2007 to 2017, lot of software tools and informatics tools has been increased, and which made awareness as well as increase the process of drug discovery. But now in 2020 and another 20 years, we can imagine that AI and ML are going to be lead the drug discovery process. So let me give brief idea about, already a lot of information has been given to you from since 4 days about the bioinformatics and cheminformatics tools. The bioinformatics, generally we try to identify gene data and protein data and pathway analysis and interactions between the pathways. Similarly, in cheminformatics, we try to understand molecular databases and virtual screening methodologies and property calculations. When you talk about property calculations, it may be like a drug likeliness property as well as ADME properties, and various properties of the molecules can be analyzed. Also, we can utilize QSAR and in silico ADME properties for the lead optimization in order to bring lead which is having more specific towards the particular protein. So we have bioinformatics databases like sequence databases and pathway databases. And in case of cheminformatics, we have already seen like small molecule databases, structure databases, like protein information, 3-dimensional structure can be found out from the Swiss port, or it may be from the PDB database. And property calculations, we have a lot of online tools are available. If you are interested, you can just go to click2drugs.com, where all the databases and all the softwares will be given at one place. So coming to the computer-aided drug design strategy. If you know the 3-dimensional structure of the protein, then you can use that as a starting point. Here just I want to give a few tips how to select the three-dimensional structure of the protein. You cannot directly take any three-dimensional structure. There are certain rules you have to follow. The first thing was the resolution of that protein. When you enter into the PDB, if you just click out any protein structure, there are a lot of entries you are going to be coming. So among that entries, which entry you have to take? The sequence remains same, structure remains same. But why there are a lot of entries? Because people might have co-crystallized that particular protein with the different small molecules. In case of COX-2, you can see COX-2 structure with rofecoxib, COX-2 with valdecoxib, COX-2 with celecoxib. So among three, which one I have to take? That will be the major query most of the people will think of. Like when you want to take out the protein structure, you have to see the resolution. The resolution should be between 0.8 to 2 angstroms. Then the co-crystal, that is nothing but the inhibitor which was present inside the protein structure should have low IC50 value. In case of like COX-2, rofecoxib, valdecoxib, and celecoxib, we have three structures. So celecoxib is having lowest IC50 value when compared to the rofecoxib and valdecoxib. So you have to take a protein structure which is having celecoxib, which contains low IC50 value. So why this low IC50 has come means when the celecoxib binds to the COX-2, the conformational changes are in a proper way. Because of that, the celecoxib was showing good IC50 or good potency when compared to the other coxibs. So you have to see the small molecule which was present as well as the resolution. So based upon that, you can select the protein structure. The another important point was there should not be any missing amino acids in the protein structure. If any missing amino acid is present in the protein structure, when you design a new molecule, you are going to be in a false positive results because the amino acid which is important for the activity is going to be missing in your structure. So that is about when you have a three-dimensional structure is available, that may be by X-ray crystallography or NMR methodology. Now you can find out some PDB entries which are from the electron magnetic studies also. So if the protein structure is not available, then you have to generate a protein model by using homology modeling technique. There are a lot of models are available and the tools are available by which you can generate the homology model. So once you find out the structure of your small molecule, either you can do docking protocol or the de novo strategy to understand the binding affinities and calculate the binding energies, or you can call it as a docking scores. So the basic idea was you are utilizing the three-dimensional structure of the protein, hence it can be called as structure-based drug design. Coming to the another methodology, if the protein structure is not available, only the small molecule information is available. So during that cases, you have a lot of information about the various inhibitors which are already present in the literature. Either they may be potent or less potent or inactive molecules, but they give some information to you that these particular molecules can be acting against particular protein structure. So during that case, you collect all the inhibitors irrespective of their potency. Then do the 2D structural similarity and substructure search between those inhibitors and find out the pros and cons of that inhibitors. Like what are the important functional groups or the core groups. Similarly, what are the negative groups which are making that particular molecule as a inactive molecule. So that information can be collected and that information will be utilized for designing a new molecule. Similarly, pharmacophore methodology, you can find out what are the common pharmacophores which are present in the molecules. Then you can implement 2D QSR and 3D QSR technologies to design new molecules. The information can be collected from the literature, journals, as well as from the patents, and wherever the authenticated databases are there, from there you can collect the information and you can design a new molecules that will be called as a ligand-based drug design methodology. So once you find out the information from the structure-based designing and ligand-based drug design methodologies about the information of protein structure and the small molecules which are already available, now you can utilize external databases. Like you have a drug bank database, zinc database, where already existing molecules are already present for various disease conditions. So by using these information from the structure-based and ligand-based discovery process, the pharmacophoric features which you find out from the ligand-based methodology or the active site which you find out from the structure-based drug design can be used as a query And you can screen all the external databases against these two features. That you are going to call it as a virtual screening technology. So here the virtual screening means in silico virtual screening. So once you screen the molecules, the next will be screening or calculating the ADME properties, which also includes drug likeliness analysis. So based upon these screening methodologies, whatever the molecules you find out from the databases, you are going to do focused high throughput screening. So here the focused high throughput screening is in vitro or wet lab experiments to find out whether those molecules are going to be showing either inhibition or activation against the particular protein structure. Once you find out certain molecules, it's not compulsory that all the molecules which you design or shortlist by virtual screening should show activity in the wet lab experiments. There are certain cases where you can find out the top hit molecule in the virtual screening becoming inactive in the in vitro experiments, because there are certain rules you have to follow when you are shortlisting the molecules. When you're talking about the docking scores, it's not compulsory that top docking score molecule has to show top activity in the wet lab experiment, because sometimes the confirmations will give you top scores, but they cannot maintain the proper interactions, which are going to be important for the conformational changes of the protein structure. So when you're shortlisting the molecules, you have to follow certain rules for the shortlisting of the molecules. Then if you follow those rules like docking score, interacting amino acids, as well as different scaffolds you have to shortlist, and these molecules when you screen against in vitro experiments, they definitely show some activity, and further you can modify that particular lead molecule, get a drug like molecule. This is about the improvement and how to utilize the computational tools in order to get a focused libraries for your particular disease condition. Coming to the body on chip technology, most of you may be knowing. So instead of doing experiments in vitro labs and with lab experiments, we can use organ on chip or body on chip methodology, where the organs or the cells which are corresponding to the particular organ will be connected through a microfluidic connectors in a single chip. Once you inject the drug molecule, it can either enter into the lung cells. So the changes in the mechanics of that particular lung cells are going to tell about the absorption of those molecules. Similarly, when you are find out the mechanics or the changes in the heart cells, it is going to tell about the contractility and conduction of that particular molecule on heart cells. Similarly, in bone, it is going to tell about the immune response. When you're talking about kidney cells, it is going to tell about transport and clearance value. So this is almost gives you the clearance of your molecule instead of doing the pharmacokinetic studies. Then when you are talking about the entry of the drug molecule into the liver cells, then you can implement and understand the metabolism of that molecule. So this is about the body on chip technology. It is costly chip. I'm not saying that it is easily available and at cheap cost, but when compared to the killing of animals and taking lot of animals and when we do the animal studies, there is a possibility that sometimes the human errors may happen. But in case of body on chip technology, at a time you are going to get information about absorption, immune response, and clearance and metabolism, so that the process becomes easy, as well as it gives at a time results to you. Most of the companies are using organ on chip and body on chip technologies if they want to screen any molecules instead of going to the animal models. This is about advances in the screening methodologies. Now let's see some case studies where I have implemented some informatics tools in order to come up with a molecules which can become potential leads. So let me explain my own study where I worked on a galactin-1 inhibitors. The galactins are a family of lectin proteins. These proteins have the affinity towards the sugars. So these proteins will be overexpressed in cancer cells and cancer conditions. So, but till now, there are only two companies who are working on a galactins, one in US and one in UK. As per my knowledge, in India, none of the company was working on this particular protein, but it is very essential protein which is upregulated in cancer. Now people have identified that this particular proteins, lectin proteins, there are 12 isoforms are available, galactin 1 to 12. They are overexpressing in various disease condition, in Alzheimer's, diabetes, rheumatoid arthritis, and Parkinson disease. So it's not that it is very new protein; it's a very old protein which was present since 1998 in the literature, but biology was explored very much, not the chemistry part. So I have selected this particular protein, and I could able to find out that only it contains carbohydrate recognition domain. As I said, these proteins are having affinity towards the sugars. So the existing molecules which are available in the literature are completely sugar based, like monosaccharide, disaccharide, which are linked by various scaffolds are the linkers. So my aim was to identify non-carbohydrate galactin inhibitor. Why? Because synthesis of the carbohydrate based drugs will be very tough process and costly process. Similarly, these are hydrolytic in nature, like when you take in the body, hydrolytic cleavage happens because of lot of hydroxyl groups which are going to be present in your sugar molecules, and stability of those molecules are very less. Hence I want to design non-carbohydrate human galactin-1 inhibitors. So we have utilized structure-based methodology and ligand-based methodology uh, to find out the non-carbohydrate molecules. Initially, we screened against natural product databases, and we could able to find out that coumarin moiety is going to fit into the active site pocket of the galactins, and we could able to find out coumarins, and which are connected with various functional groups, hydrogens and pyrazole and benzothiazines. So various permutations and combinations we have taken, and we try to design a small molecule which was completely non-carbohydrate in nature. So luckily, when we screened those molecules which are feasible to synthesize, and these molecules we tested for galactin-1 enzymatic inhibition assay, and we could able to find out that these molecules are reducing the galactin-1 protein levels in the cancerous cells. And after that, we want to find out whether they are really altering the galactin function or not. Hence, we performed fluorescence spectral studies to understand how the binding affinity is going to be happening, because the main aim was the molecule should have association and dissociation, then only it can able to show the function. So when we did the fluorescence spectra studies, it is going to show the binding affinity, and further we have taken those molecules and screened various apoptotic parameters to understand whether they are really going to be anti-cancer in nature or not. So that is one part, and approximately we have synthesized 120 molecules, combinations of coumarin and benzodiazoles and various linkers. And these molecules, luckily, 8 molecules were showing good anti-cancer activity, where they are having affinity towards the galactin-1 protein, as well as some of the molecules we have taken for the biomarkers, radio-labeled biomarkers, to find out the disease condition of the cancer. Similarly, that is about the more focused on the cancer activities. Next, we have done some of the molecules we have studied, surface plasma resonance studies, where exactly these molecules are, how they are altering the function or the binding affinity of these molecules. So we have done ADME properties and toxicology, molecular dynamic simulations, and we could able to show that these non-carbohydrate molecules also having affinity similar to the carbohydrate type of molecules. So this complete project was funded by the DBT. So the next part was we have implemented 3D field-based QSR approaches. Why I'm giving this case study was you can design the studies in a similar fashion to come up with a good conclusions. So instead of purely depending upon the structure-based drug discovery, we have utilized ligand-based drug discovery. So as I said, there are a lot of galactin inhibitors are available, and all these are sugar based. You could see like the monosaccharide and disaccharide linked with various linkers, as well as some are peptidomimetic molecules which are available in the literature. So we have collected all the known 324 galactin inhibitors which are of different saccharides. Also similarly we have collected 52 inhibitors which are monosaccharides. So we want to find out whether monosaccharide is important for the activity or the disaccharide is important for the activity. So we performed 3D field based QSAR study where the galactin inhibitors has been collected, and here the Ki values or the Kd values you have to collect. So most of the time we will misunderstand that QSR can be performed for the newly synthesized molecules without the biological activity, but that is not the case. You require the biological activity when you want to perform the QSR studies. When you design and synthesize 20 molecules or 30 molecules, you can perform QSR, provided if you have a protein inhibition data or at least you have MIC values or any IC50 values. You require biological activity data if you want to do the QSAR studies. Once you collect the molecules, like there is a protocol by the Schrodinger QSAR model where you have to convert into 3D structures and convert these IC50 values into PK values. Once you collect the molecules, you divide them into training set and test set. The optimum methodology was 70 into 30. So once you divide the molecules, because you have to first train the QSAR model, then you test the QSAR model, whether your model is going to be correct or not. So here you are going to implement partially square method statistical model. Once you do the modeling, you will get a scatter plots which tells about the linearity between the training set and test set. Then you can do the visualization. This is simple protocol. So when we perform the 324 ligands, we could able to see two graphs. One is for the training set, other is for the test set. When you do the QSR, you will get a lot of statistical parameters like standard deviation, R square, stability, F value, Q square. So you have to find out the model which is having a best R square and Q square values and highest F value. Like that, there are certain rules you will follow to shortlist your model. So similarly, in case of 52 ligands, we could able to find out another statistical model. You can see when you see this particular graph, the dots represent the molecules, the training set molecules, 70 molecules, how they are near to that particular line. So if they are away from the line, means they are deviating certain property. Similarly, in case of 52 ligands, most of the molecules inside the line. So from this, we found out that there are certain parameters which are important for the molecules to have affinity towards the galactin. So instead of comparing all the molecules, we have selected high potency molecule, moderate potency molecule, and low potency molecule. And we try to find out the 3D field bases which are important for the activity. So first one was the steric features. You could able to see the round one where I have just circled as a red, that is absent in case of low potency molecule, whereas that is present in the high potency molecule. That could itself will explain you that steric feature is very important to show the high potency. Similarly, hydrophobicity, where I have circled it as a blue color, the hydrophobicity part is absent in case of low potency molecule, whereas in case of moderate and highly potent molecule, this hydrophobic property is present. Similarly, hydrogen bond acceptors and donors, where I have circled it as a red color in case of all the figures. In case of low potency molecule, that particular hydrogen bond acceptor is missing, whereas that is present in case of high potency. In case of donors also, similar fashion. Coming to the 52 molecules, that is monosaccharides. Instead of comparing disaccharides and monosaccharides, so in case of monosaccharides itself, with different ring systems which are connected with the monosaccharides, the steric feature which was missing in a low potency molecule, whereas it is present in the high potency molecule. So for even in monosaccharide, the molecule can be active, provided it should have that particular steric feature present at that particular position. Even in case of hydrophobicity, as well as hydrogen bond donor and acceptor, we have compared and we have come up with a conclusion. Before concluding this features, 3D field based features, which can be important for the biological activity of the galactins, we have to validate the model. It's not that these results may be correct or incorrect. So we have validated the model by using external validation, where you have to calculate various properties like positive errors, positive negatives. So these statistical models, once you understand or learn the QSAR completely, how you are going to dividing training and test set. From the training set, how many are actives has been predicted as actives, and how many inactives has been predicted as inactives. So such type of protocols you have to follow and find out whether the model is validated either by internal molecules or the external molecules. Here the internal molecules means you have collected 324. Among that, 70 I made it as a training set and 30 I made it as a test set. So that is nothing but your own model, your own molecules which are known to be as a galactin inhibitors. But in certain cases, you can use some other molecules which are not at all related to galactin, maybe some other anti-diabetic drugs, and you can validate against this particular protein. So such type of external validations and internal validations you have to perform for your QSAR model and find out whether they are within the limits or not. For doing these calculations, we have used DTC labs, which is the QSAR protocols and the QSAR models softwares are available with Dr. Kunal Rai. You can find out from his website. They are freely available. So after validation of our model, we have concluded that for galactin inhibition, we require aromatic substitution at that particular position, as well as ester linkage is unfavorable and the aromatic amide linkage is favorable at that particular position. Similarly, there should be some aromatic ring system which should be present for the steric favors group. So like we could able to find out what are the important features before designing a molecule. Based upon this idea, we could able to design sugar based molecules as well as non-sugar based molecules which could able to show the galactin inhibition. And this work has been published already in informatics in medicine unlocked journal. And based upon this, we have synthesized 10 molecules, and all the 10 molecules were showing galactin inhibition. So next was to understand how the galactin inhibitors will be helpful in the coronaviruses, because when the corona started, we thought that the galactins we can show for the coronaviruses, because the coronavirus is having carbohydrate recognition binding sites in the spike protein. So briefly, I just would like to tell you that these coronaviruses have carbohydrate recognition domains, which are structurally similar to the galactins, and they bind to the ACE2 inhibitor before entering into the host cell. So they require this CRD, that is nothing but carbohydrate recognition domain, to enter into the host cell. And we know that hesperidin is a glycosylated ligand which binds to the SARS-CoV-2 ACE2 RBD domain and disrupt the interaction. So based upon this simple logic, we started exploring how the galactin inhibitors are going to be effective in the corona disease condition. So for this, I'm not going in depth. So we have taken a spike protein, and all the 330 galactin inhibitors which I have shown in the earlier study has been screened against the spike protein. So here the important point was in spike protein, there are a lot of binding sites have already been reported. So we have done binding site analysis and selected the site where the ACE2 is going to be binding, and we performed the docking studies. After that, we performed molecular dynamic simulations and molecular mechanics simulations, binding affinity simulations between the molecules which were showing good affinity towards the spike protein. So these are the two molecules, 213 and TD139. In the last corner of your screen, you can see these are the two molecules which were showing good binding affinity and good interactions basically to the spike protein. And these two small molecules we screened by doing molecular dynamic simulations, as well as ADME properties, toxicity properties, and not the drug likeliness properties, because these molecules are sugar based. They cannot follow the drug likeliness properties. They are more than log-P will be very high when compared to the other molecules. But already these are in the literature. We have not synthesized any new molecules. And this TD139 and 213, these two molecules are already present in the clinical trials for the NASH disease conditions. So from this study, we concluded that this TD139 is having acceptable ADME properties when compared to the 213. We have concluded TD139 can become a one of the best molecule which can target the CRD domain in the spike protein. So as soon as we just submitted this publication for the journal, we could able to see there was one post from the clinicaltrials.government in July 16, 2020. They just said that this particular molecule TD139, which was by the Galactin Therapeutics Limited company in US, is entered into the phase 2 and phase 3 clinical trials for the COVID-19 disease condition. As I said, this molecule is already present for the NASH disease condition in the clinical trials by the same company. They have taken for the COVID-19 treatment. So we can say that the experiments or the in silico conclusions, whichever we have driven from the spike protein interaction with the TD139, can be making this molecule to act against the COVID-19, because this was the paper we published in the journal in the month of July, same month when the molecule has been entered into the clinical trials. So this is how, when you do the proper interpretations of your studies, in silico studies, you can have proper in vitro results also. So there should be conclusions which are going to be driven, the combination or correlation between the in silico to the in vitro experiments. So next, another case study which I was just started in collaboration with Institute of Genetics. They come up with a problem stating that some of the people are not responding to the salbutamol. When they come for the asthma disease duration, generally the doctor will prescribe salbutamol. So they said that some of the population are not responding to the salbutamol. Then we have performed some in silico studies. There are already reported mutations which are going to be present in the beta-2 adrenergic receptors. As we know that salbutamol binds to the beta-2 adrenergic receptors, and this is very good beta-2 agonist, salbutamol. So we just did some literature search, what are the mutations which are present in the beta-2 adrenoreceptor. We could able to find out there are 3 to 4 mutations are present in that particular receptor. But among that 4 mutations, which mutation is placing a major role so that the salbutamol is not responded by the many patients? So we performed in silico studies, and we could able to find out threonine 164 to isoleucine, which was I just rounded up in the first figure. That particular mutation was very near to the agonist binding site. And when the agonist binds to this mutated protein, you can able to see there is a lot of variation in the protein structure. In the second figure in the bottom, you can see there is a red color helix is present, whereas in the mutated protein, that helix has become a loop. Because of that, the three-dimensional structure of the protein is completely changing at the active site. And that may be one of the reason why the salbutamol is not binding to the beta-2 adrenoreceptors in case of mutated patients, like threonine to isoleucine mutation. And there are a lot of hydrogen bonds and hydrophobic interactions are going to be changing between the wild type and mutated protein. So this study has given and enlightened that this mutation, if the mutation is present, that particular person may not respond to the salbutamol. So the doctor has to prescribe some other medicine. So that was the conclusion which we generated from the in silico studies. So from that time, when the patient comes for the asthma treatment, they started screening the mutation. If the mutation is there, then they are directly going to other alternative drug for the asthma. Similarly, other aim was to find out a molecule which can bind to the wild type and mutated type protein. So we have performed in silico virtual screening by using structure based and ligand based. As I discussed with you in the previous slides, we have selected various databases like the databases, SNX database, drug bank database, zinc database, natural product database, lot of databases we have selected, and you performed side by side ligand based and structure based drug discovery, and we could able to find out a single molecule which I represented in a box. That molecule was showing good affinity towards the wild type and mutated type protein, and further this molecule we have taken for the in vitro experiments, like reducing the cGMP levels as well as increasing the beta 2 adrenoreceptor mediated uh, intermediates, and this is showing good activity. The results are still going. So this is about how the in silico work can be taken towards the in vitro experiments, and you can have there is proper information about the in silico to in vitro studies. So this is about informatics. I think lot of informatics we have seen now. Let's we move into the machine learning models. The main aim of the conference was AI and ML and deep learning models. Let me explain what is the major difference between these three models. So in previous cases, we have seen that we have utilized a lot of information tools and the software tools to do the discovery process. So in case of artificial intelligence, this is nothing but like I can say it's a broad view of the deep learning, where the ability of the machines to simulate human behavior and take decisions intelligently like a human. So the computers can take a decision, but at each stage you have to define your conclusions. Like if first step has been done in docking, like protein preparation, all of you knowing. So once the protein preparation is done, then you have to explain the model that whether you have to take forward or the backward or yes or no conditions. At each stage, you have to guide the computer to take a decision. But in case of machine learning models, it was computers automatically learn from the given data. Once you fix the program, it can able to understand the conclusion from that particular program. In deep learning, it doesn't require any extra information. The computers can directly take the decisions. It doesn't require any training of your computer. You have to just input the data, and data can be analyzed by own, and it can give you the results. This is the major difference between the AI and ML and deep learning methods. Let's see which type of data we have to give to the computers. So there are varieties of data we generate during the experimental models. So in case of data, when we are talking, there are two varieties of data. Excuse me, madam, how much time I have?
Hello, Malika. Am I
Yeah.
audible?
Yeah, how much time I have?
I think we can continue this session up to 11:30, maybe 20 minutes more.
Okay, yeah, thank you. So different types of data we will generate during the experiments, and depending upon the data, we have to implement different models. So you have qualitative data and quantitative data. Qualitative data will be categorical in nature. Categorical is nothing but it can be a text or a string, like yes or no conditions. When you're talking about any experiment, uh, the BP has been raised or decreased, yes or no conditions. There is no numerical values are involving in the categorical data. But in case of quantitative data, you have a numerical data where certain numbers are going to be present, top priorities. So when you're talking about qualitative data, there are two types. One is nominal data and ordinal data. This slide you can find out in the Google itself also. And nominal data will be having no order to it. No order means it may be like gender, hair color, or ethnicity, like woman or man, certain only two different conditions. Whereas in case of ordinal data, there is a order, like first and second and third order, and the letters like ABC grades when you are giving for the experiments. Similarly, in quantitative data, as I said, it is a numerical data. You have discrete data and continuous data. Discrete data means they have a logical end to them. So the number of students in a class as well as number of workers in a company. So these things will be certain numbers. Like in a class, you have 40 students. So the 40 student is the intake of the class. Like that, there will be a logic end to them. Whereas in continuous data, there is no logic. Like the height of the children. When you see the height of the children, it increases, and it depends upon the age and weight of the children. The height is going to be changing. So you cannot fix that there will be only 10 years baby should have only this much height. So that type of data, we call it as a continuous data. There is no logical end, but they keep increasing or decreasing depending upon the nature of that particular property. So these are varieties of data you have to handle when you want to do the AI and ML technologies. So the first important thing is which type of data you are handling, that you have to decide. Depending upon that, the statistical models can be implemented. The next method, next important thing was the how to split the data in the machine learning. Whenever we do the machine learning models, the important thing was, as like QSAR, you have to train your data. That means you have to train your computer with the certain data. Again, you validate the data, then do the testing. So these are the three steps we do when you do the machine learning models. So the properties of data include the 5 V's, which is volume, variety, velocity, value, veracity. These are the 5 different parameters that also you have to check it out when you want to perform the ML models. So coming to what is difference between the machine learning and the deep learning models. So the simply I'll explain. Machine learning model is nothing but the input. As I said, you any type of data you can give as an input. It may be categorical data, continuous data, or numerical data, anything that may be in the image form, text form, or table form. So that input is nothing but any image or any data whichever you are giving or feeding into the computers is try to extract the features. For example, if you're talking about this particular image, what is the size of that tumor, and what is the blood vessels which are going to be present, what is the thickness, blood vessels. So all these features you have to extract. And there is one important function which is going to be present in the machine learning is the classifier. So this classifier try to understand the relation between these features. So when you're giving the input as 100 inputs, 100 images with different features, like having similar type of features, this classifier try to find out the connectivity between the blood vessels and thickness of the blood vessel, and the tumor size, as well as the how many blood vessels are present. So this classifier plays a major role to find out the connectivity between the features. Based upon that classifier data, whenever you give a new input, so already your machine already having certain conclusions or the in-depth calculations which are going to be present from the training data. When you give a test data, it will try to analyze what is the blood vessel size and the image size and the depth of the thickness of the blood vessels, and it is going to tell you whether it is a benign tumor or a malignant tumor. But in case of machine learning, first of all, you have to define the output also, which type of output you are expecting. So based upon these properties, you want to just give only two answers, whether it is a benign or malignant tumor. But in case of deep learning, when you give the input, feature extraction and the classifier is done by the computer itself. Now here the human cannot give the feature extraction. As I said, computers are going to do everything in the deep learning. So based upon the image itself, as you feed all the data, that data will be taken, and classifier also will segregate the data and gives you output as a benign tumor or a malignant tumor. This is a major difference between the machine learning and deep learning. In machine learning, there are two steps in between the input and output, that is feature extraction and classifier. But in case of deep learning, feature extraction and classifier is a single job which will be done by the computers itself. So let's see what are the different varieties of the machine learning models are present. As I said, machine learning or the AI and deep learning, all are statistical methods. These statistical methods we used to utilize in the QSAR studies and many other experiments. But there we used to handle limited data. When you want to utilize these statistical models for the huge amount of data. We call it as a machine learning models, where the machines can try to analyze the data and give you the inputs. So we have supervised learning, unsupervised learning, and reinforcement learning models. So in case of supervised learning, it is a task driven. That means what type of task you want to do. Whereas in case of unsupervised learning, it is a data driven. As I said you, in unsupervised learning, it only gives you the correlation between the data. Like if you are having two different sets of molecules in your data set, it just segregates the data into like the molecules which are having high log P and low log P. Like that, it is data driven. In case of reinforcement learning, it is an algorithm learns to react to an environment. It's like LIC agent, like he used to come every time and he try to find out the people's opinion and go back and again modifies the company's profiles. So it's like that reinforcement. It tries to validate the model by itself and come up with a new improved results. So in case of supervised learning, you have various models. One is regression model, decision tree, random forest. That in case of continuous data. You can see here two segregations I made, continuous data and categorical data. In continuous data, you can implement these three models. Whenever you have a continuous data, then you can implement regression, decision tree, and random forest. In case of unsupervised learning, if you have a continuous data, you have to use clustering models. Similarly, if you have a categorical data with you, then you have to implement classification models as well as association analysis and hidden Markov models. So as I said you, first important thing was which type of data you are handling. Based upon data, you have to implement the model as well as the statistical test to get a results. So this is a brief about the machine learning models and how different varieties can be implemented. So coming to the supervised learning, what exactly it is. So I'll just explain you in a very basic understanding level. If you ask a baby by giving two fruits to her, like banana and apple, so how she remembers. So she may not be knowing at the first time this is apple and banana. She just imagines that by seeing the image itself, the round shape will be apple and non-round shape will be the banana. Similarly, the red color will be apple and the yellow color will be banana. That was the imagination the baby can have without knowing the name. So you are going to give the features. As I said, in machine learning, you have to give the features. For apple, you are going to give the features like it is round, smooth, and red color. In case of orange, it is round, bumpy, and yellowish color. Then in banana, it is curved or not round. You can say it as smooth and yellow. So these are the features you are going to define for each fruit. As I said, I'm going to give the basic explanation. This can be implemented for any field. So the next stage was the classifier. So what this classifier is going to do, first query was whether it is round. So whatever the features you are defining in the feature extraction, the first feature was shape. So first query was whether it is round or not. So if it is round, then it is going to say that whether it may be apple or orange, or else it is going to be a banana. So the next query was here it is orange, it has been written, but it should not be written that it is a bumpy or smoothy nature. So in orange it is bumpy, and in case of apple it is a smooth. So the next query was it is bumpy or smooth. So like that, the computer is able to find out the classifier from the questions which you have or the features which you have defined in the feature extraction. So the next time when you are giving a new fruit, you can see this is a banana, but not in yellow color. So what is the output it is going to give us? So the output will be banana. Why? Because the first query itself, whether it is round or not. So if it is not round, then obviously it is going to give you as a banana instead of going to the second query itself. So similar type of protocol will be followed in case of the benign tumors and malignant tumors, where you are going to give lot of images which has been produced by the CT scans, and during the image feeding itself, you are going to give the features which are present in the image. So whenever you give a new image, this new image will be screened for all these features which you have defined in the training set, and it is going to give you whether it is a benign tumor or a malignant tumor. Coming to the unsupervised learning, as I said you, it is a data driven. It's not a task driven. In case of supervised learning, you have an output, whether it is a banana or orange or apple. But in case of unsupervised learning, it only gives you or try to give the conclusions very easily by segregating the data. So you have a baby and showing him that 5 animals. He doesn't know whether it is a dog or cat or anything, but he can imagine by seeing the picture itself that there are 3 animals which are taller and 2 are smaller. Okay. So when you ask this boy what are these animals, he can able to say that there are 2 types of clusters. 3 are of same height and 2 are of low height. So unsupervised learning try to segregate your data depending upon the parameters. It may be single parameter or various parameters. So in case of small molecules, if you take hundreds of molecules with you, when you perform unsupervised learning, it try to segregate your molecules into 3 clusters. One cluster may be having high molecular weight, or another cluster may be based upon the log P, and another cluster of molecules based upon the hydrogen bond donors. So by clustering, you can understand that how many molecules are having similar type of features. Among this, how many are active or inactive against a particular protein condition. So this is how the unsupervised learning works. This is very basic explanation about supervised learning and unsupervised learning. And these are the methods which I have shown in the previous slides about the machine learning models. As I said, all these are statistical models. Before machine learning also, we used to utilize these models, but not for the huge data. But the function and formulas and the principle behind the models remain same. Only the data where you are applying these models will be different in case of machine learning and AI models. So as I said, we used to develop lot of images by using experiments. So at each stage of the drug discovery process, like target identification, we used to generate functional data, genomic data, and proteomic data in the form of images. In lead identification, by high throughput screening and microarrays testing, we generate the images. And lead optimization, like SAR development and ADME methodologies, animal images as well as SEM-TEM images. And during the animal testing and clinical testing, we generate a lot of information. But among this information, only limited information we used to utilize and conclude. There is no effective way to mine the image data. The numerical data can be mined very easily, but image data cannot be captured very easily. So during that case, this AI models has given lot of scope to analyze the images in depth and give the connectivity or correlation between the various parameters from one stage to the another stage also. So let me come into the natural product, because most of us work on the natural products. As is came to know that some of the queries we have seen from previous sessions, that natural products. So it's not that only the machine learning can be implemented in the chemistry or any other models. From natural products and natural extracts also can be identified by using the machine learning model. This is one of the interesting paper where Reher et al. Has reported NMR based machine learning tool that is called as SMART for structural analysis of major constituents from the crude natural extracts. So here actually what he did was, he just the new NMR data has been collected and connected to the machine learning model. Like the data which he generated from the NMR of the various natural products has been analyzed by the machine learning tools and try to find out or characterize a new molecule, which is a macrolide symplocolide A, which is from the cyanobacterial extract mixture. So machine learning can be implemented even in the natural extracts isolation also. Similarly, another paper where natural extracts has been identified was the finding out the function of the small molecule based upon the 2D structure. Till now, based upon the 3D structure, we could able to find out where it is going to be binding and everything. But based upon the 2D structure, by using artificial intelligence, Liu et al. Has given a good protocol. Like this paper talks about, he has taken all the known natural products and the different PDB entries, like different PDB structure, whichever he is interested, maybe different proteins. So he tried to implement the machine learning model, like collecting all the natural products and different properties or the scaffolds which are present in the natural products has been collected, and how they are going to be binding to that particular different proteins has been studied. So he has just collected the fragments and he defined a classifier, could able to find out the binding affinities and what are the off targets and competitive or non-competitive inhibition. So it's very simple that he has just taken a natural products and divided the molecule into different fragments, and these fragments has been studied against various proteins by using docking calculations or any other calculations, and could able to find out this particular fragment is having affinity towards the COX-2 protein. So he can say that this particular uh, natural product can be acting as a COX-2 inhibitor. So this was the predictions he made, like he predicted the functions of new compounds, also fragments to get a new molecules, and predicted the some side effects also. If some fragment is having affinity towards the COX-2 as well as some other protein, you can say that some other property may become a side effect also. So this was the protocol which was followed in natural products. So let me explain what are the things you should have when you want to do machine learning models, or if you want to learn machine learning tools. So as I said, irrespective of whether you are a computer programmer or not, you require little programming language skills if you want to perform machine learning models. The first thing was the Python. People can utilize R language or SQL language or C++ or C, but emerging was the Python, which is user friendly. If you know programming in the Python, you can easily do the machine learning. So not only the programming, you should understand how to handle the data. As I said, if you have images, if you have tables, if you have Excel sheet data, or if you have arrays of data, then you require NumPy and Pandas are the data which can handle the numbers. Whereas Matplot library and Seaborn, these two will be utilized to visualize the data which you have used in the model. So whenever we do the Excel sheet, when we want to plot a graph x-axis and y-axis, we just go to Excel sheet and graph the plot. Similarly, by using Python, if you want to plot a graph, you have to learn how to do the representation in different varieties of graphs. You may be showing bar graphs or any other graph models. You have to utilize these two packages, Matplot library and Seaborn, which help us to visualize the data in different forms. And coming to the chemistry point of view, if you want to calculate any chemistry properties or the properties of the molecule, like molecular weight, total polar surface area, or any other properties, Wiener index, so you have to call RDKit and ChemPy. These are the two programs which can help us in Python to calculate various properties. All these for Python, NumPy, Matplot library, and RDKit and ChemPy are the basic programming languages as well as the packages. But main important thing was the ML packages: scikit-learn, TensorFlow, Keras, and Spark. These are the four programs where they help us to implement the statistical models. Like if I want to have a simple linear regression where you have a single independent x-axis and y-axis. But when you want to do multiple linear regression, you have multiple independent variables and single dependent variable. So there are various formulas which are going to be present when you want to calculate y is equal to mx plus c in case of simple linear regression and multiple linear regression. So instead of writing each formula every time, this scikit-learn and TensorFlow, Keras, and Spark have inbuilt packages. Like whenever I want to do the simple linear regression, it's not that compulsory I have to write the formulas. You just call a program or a package called as linear regression. Then all the formulas related to that package will come into your program sheet, and they can calculate the R-square values and whatever you want to calculate by using that particular equation. So you require a little bit of programming language, and Python is very easy to learn. If you know simple mathematics, if else conditions, yes or no conditions, how to represent subtraction, addition, like conditions. So these are the programming languages and packages you have to learn before you are going into the machine learning implementation. So with this, I just would like to conclude my session stating that drug designing is an art, but applications of informatics resources and data analytics will play a major role in the attempts to make it more rational and successful in the future. as I discussed from the first slide onwards, like the decisions whichever you take by using various information tools is going to place a major role in the success of your drug discovery process. And before ending my session, I just would like to introduce my Mass Training Academy, which was very budding academy, I can say, established on 3rd April. And I'm going to provide all the online classes in case of fundamentals of CAD and hands-on session. In hands-on session, I'm going to give all online tools which are freely available, not commercial, and Python and machine learning also. And successfully, we have completed 4 batches, and more than 200 students and faculty all over the world has been enrolled and successfully completed the course. And these are the some of the universities from where the students has been enrolled for the courses, and they successfully satisfied with the training which they got from the Mass Training Academy. And we have every month 1st and 15th new batch is going to start. So thank you one and all for patient listening.
Thank you, Mallika, for your elaborate continuous lecture for more than 1.5 hours. I think now we will break for 10 to 15 minutes, then we will join back for your demo versions.
Yes, definitely.
Just a second, ma'am. There are some questions.
Pooja, we will handle the questions at the end of the session, because now if we start taking questions, the session will continue and we will not be able to break.
అయిపోతుంది సెషన్ నేనే చేస్తా.
So participants will break for 10 minutes. In 10 minutes, we will join back. Then Mallika madam will be continuing her presentation. And all the questions we will handle in the last. Is that okay, Mallika? Is that
Yeah,
okay?
yeah, fine, fine. No issue.
Okay, we'll handle the questions at the last.
Yeah, yeah. Thank you.
Okay, let us break for 10 minutes. Hello.
Hello, ma'am.
Shall we start?
Yes, yes, yeah. Yeah, my slides are visible.
Visible. They are visible.
Okay, yeah. Let me just give a brief idea about how we are going to do the hands-on session. Then I can start the session. Can I start now?
You can start.
Yeah. So as I explained you about AI and ML models, and it is going to have various packages and programming languages which are important to learn, as well as we should know how to write a script. So let me explain the same example like orange and apple, which I explained in the first session. So when you have apple, that will be the features which are going to define are going to be the features, and the classifier will try to analyze the data and gives you the output. The same principle has been implemented when you have a mail segregation. Some of the mails goes into the spam. So the programmer, the Yahoo or Gmail programmer might have written some words, keywords. If the keywords are going to be present in your mail, that mail is going to spam. So the feature and classifier plays a major role for segregating the data. So in today's example and today's hands-on session, I'm going to explain you where exactly you are going to write the programming. That is very important to learn before going into the actual modeling. So I'm going to explain about simple protocol. Then I'll show you an hands-on where you are going to do these experiments or these programs. So for example, you have 4 fruits with you, which are having a weight around 150 grams, 170, 130 and 140 grams and different textures. I'm going to take only 2 features. One is weight and texture. It's not, there is no any fundamental rule that you have to take only 2 or 3 features. You can have n number of features with you. But you should have features as well as label. Label is nothing but the output. So once you train the program or the computer, you are going to get a output that you call it as a label. So the weight of the fruits was around 130 to 170 and texture was 2 textures. One is bumpy and smooth, and bumpy texture I'm going to give it as a label as orange, and smooth texture I'm going to define it as a apple. So if you want to write your program by using these 2 features and 2 labels. So the decision tree, how it is exactly working. So once you give the features, the decision tree comes into the picture. So it first have a question like stating that what is the weight of your fruit. If it is greater than 150 grams, yes and no conditions. If it is yes, then it is going to have another query. What is the texture, whether it is bumpy or not. Based upon this bumpy nature, you can get apple or orange as output. If it is like in previous case, I have given it is a round shape or a no round shape. So this is the decision tree which plays a major role. So for doing any program of machine learning, you require features. First, you have a features with you and the labels with you. Once this type of data is available, you are ready to write the ML program. So how I'm going to write the program? I just made the script over here. I'll show you again in the hands-on also. So as I said you, scikit-learn is the package where you are going to do the machine learning. So from sklearn import tree. This is the statement you have to give whenever you are calling the scikit-learn program and you are importing tree. Tree is nothing but decision tree. So these are the 2 packages you have to call from the scikit-learn program, and you have to define the features. Features I'm going to define in the form of why I said Python is very important. Whenever you are defining the features of your data, you have to put it in the form of various models, like there are list, there are tuples, dictionaries, like various type of data representation is present in Python. So here the features has been represented. Features has been represented in the form of weight and the texture. Weight is 140, texture is Am I audible?
Now you are audible.
Okay. Just a minute, ma'am. I'll just join within 2 minutes.
Okay, fine. Mallika, I have a small request for you. Please make Pooja as a host. By mistake, it has changed. Only you can do it.
Yeah. Unmute.
Ma'am.
Pooja, is it okay?
Yes, ma'am. No, it is not made as host, ma'am, but I can speak. Just right click on my name, ma'am. You'll have
Okay.
option.
Yeah.
And there you'll get the option.
I'm just finding your name.
In the long list, you have to check.
Yeah. Fine.
Yes.
So slides are visible.
Yes, ma'am.
Okay.
Thank you.
So we are going to take the data, which is two features, weight and texture, and label it as orange and apple. And decision tree is going to make its own decision tree based upon the features like weight and texture. So here, how to write the code is from sklearn import tree. This particular statement you have to give whenever you are writing the code. And features we have to define in the form of list, like first parameter in the list was weight, and second is the bumpy or smoothy nature. And labels also again as a list, that is apple and orange. The first parameter feature 140,1 corresponds to apple, 130,1 corresponds to again apple, 150,0 that is bumpy nature is orange, and 170 and 0 is orange. Now as we are calling the tree as from the sklearn, CLF is nothing but classifier that you have to define tree.decision tree classifier. So from the decision tree, it is going to generate a classifier. So this particular statement you have to give whenever you are using the decision tree classifier. Then classifier equals to classifier.fit. So you are going to make a comparison between the features and the labels. That is nothing but you can call it as a fit table. or fit graph it is going to generate by its own between the features and labels. So now I would like to explain you where you are going to write this particular program. So whatever we are going to write, we have to do in the Python. So there are various online tools or online packages are available. You can just go to python.org, and here you download your Python package, install in your system. Okay, that is one option. Once you install the Python, you will get a window where you can write the script. Or if you are not interested to install anything, you can do online, where that package is called as Jupyter. Jupyter is online web source application for doing the programming. Hope this is visible to everyone. Sujata ma'am, is it visible? My screen is visible?
Yes, madam, visible.
Okay. So in Jupyter, this also, if you want to install permanently, you can install, or you can try online. So once you go to Jupyter, you just type try Jupyter. You have various varieties are available: Classic Notebook, Jupyter Lab, and Jupyter with Julia. Various options are available, but try with Classic Notebook as a basic programmer. It takes 2 to 3 seconds because it's an online version everybody uses. So this is how the Jupyter window looks. You have various options like file, edit, view, insert, cell. It's almost like your Word document options. When you say file, you have new notebook, open, make a copy, save as, download. So when you want to write a program, you have to open your own new notebook. This, as I said, this is an online Python like portal where you can write the Python script. So this Python 3 portal you have to open, go to file, new notebook, and Python 3. If you already written and saved, then you can open, and you can have save as option, rename option, print preview options. Similarly in edit, you have options like cut, copy, paste cells. Then in case of view, you have a toggle header and line numbers. So let's see how the window looks. File, new notebook, and Python 3. So as soon as you say that new notebook, it is going to open a new window where you have various options below the file menu bar. Save, then addition, insert the cells. So whatever you want to write, that you are going to write in this particular cell. For example, here I have just written hello world. Okay, so this is what we write in the program. In this particular given line, you have to write the program, which will be followed by the arrow symbol. Arrow represents that can be executable. Okay, then you have inserting the cells, like you can have more number of cells, you can delete the cells, then make a copy, then you have pasting option, up and down arrows. Once you write the program, if something has to be first executed, then you can move into up or move into down. Then you have a run options. So most important thing was here the code, markdown, raw, NB convert, and heading. Code means which can be executable and which can gives you the results. Whereas markdown is nothing but the statements which you write before running the code. Like for example, if I want to calculate the molecular weight, I just write a statement saying that heading is like calculation of molecular weight. That cannot be executable. That is something notes you are writing for your own purpose. So when I say markdown, you can see the changes happened in the cell. There won't be any arrow symbol because this hello world will be just a statement which was written for my own purpose. But when I made it as a code, it is executable code. So these are two important things. And then when you say heading, it becomes a heading for your particular program. So that can also be not executable. So whenever you are writing a code, you can use it as a code markdown. In between, you can make it as a headings also for saving purpose. So let's explain you first simple Python. What is Python? Hope all of you are new to the Python. So I just say A equals to, as I said you, Python follows certain format. Whenever I'm using string, that is text, you have to put it in inverted commas and run it. Then say A, print A. Now you see the answer output is hello world. So this is very user friendly. Whatever you are going to write the program, that can be executable and gives you the answer. So for example, if I say A equals to 2, then again print A. Then the output will be 2. Simple example I'll give you. A is equal to B is equal to 4 and I want A plus B. Okay, and print C. You can see the answer is 6. So this is simple Python, how we are going to write the scripts. But these are very simple examples. But when you want to have large data with you, you require some representations. As we have seen in the previous PPT, how the weight and labels has been implemented over. So let me just explain you how the initial RD kit will be used. Then we will move on to the machine learning model. So I'm going to just copy the script. So from sklearn import tree. So if I just remove this sklearn from tree, your remaining 4 commands will not be executable because first of all you have to call the package sklearn. And features are 140,1, that is one apple weight and texture. Then another apple weight and texture, orange and orange. Similarly, the labels I have given. So this Python is user friendly. Whenever you do a mistake, it gives you the error messages to you. So as I said, I have copy pasted. The comma symbol was different. So you have to make it as a understandable language to the Python. So now I have just written from sklearn import tree, features and labels and classifier. Now I'm just running the program. So you can see when I started running this program, the star symbol shows that it is running. If the number comes, means it has been completed. Run is completed. Now your program is over, stating that with features and labels with the classifier. Now if you want to predict new model. So I'm going to write classify predict. Classifier is having another package or another command, predict command. And prediction, you are giving the weight of the fruit was 160 and 0 is, as I said, it is a bumpy in nature. So what is that fruit? Again run it. Then you can say it is going to tell it as a orange. You have not given or you have not stated that particular weight and the feature in the features, but the classifier has made a table itself and stating that if the weight is 160 and bumpy in nature, that is going to be your orange fruit. Similarly, if you want to predict something having weight around 135 grams. Okay, and you are having smooth in nature, then it is going to predict and give you as a apple. So this is how we are going to write machine learning programs with simple features and labels. So hope this is how the people will write. First important thing was you require collection of data and what are the features which are going to be present in your data and what are the labels you are expecting from the data. So once these two are available with you, you can implement any type of supervised learning or unsupervised learning models for your machine learning model. So let me show you demo related to the chemistry related work. So RTKit is a chemistry related cheminformatics tools calculation. So for this RDKit, again you have to call the package RDKit. So whenever you are using Python scripts, you have to call the packages. Python itself will not do all the works. It takes the packages like NumPy, SciPy, Pandas, Matplot library, Seaborn, RDKit, ChemPy, scikit-learn. So whenever you want to calculate particular related work, you have to call that package into the Jupyter window. Then only you can able to calculate. So let's see how this visualization can be done. So if you want to visualize any small molecule, you require some visualization tools. Either you require ChemDraw or Marvin Sketch, Argus Lab, some software you require for the visualization. So if you want to visualize any small molecule without any software or visualization tool, you can use RDKit package where you can do the calculations. So let me show you how these packages will work. I'm not typing, I'm just copy pasting because it takes time to again write everything. I have already written in my PPT. So from import RDKit. So whatever the packages you want to import, first you say import RDKit. If you want to import Matplot library, you say import Matplot library. So in RDKit, various packages are again, sub packages are available. You just go to Google and say RDKit. It is going to give you all the packages which are available inside that particular program. So again from RDKit, import Chem. Chem is a package, chemistry package. Then from rdkit.com, import draw. It doesn't only require to calculate. For calculation, first it has to be converted or you have to draw the molecule. So you have to call the package draw package into the Python. Again from rdkit.chem.draw, import IPython console. As I said, Jupyter is a online Python. Even in RDKit, again you have to call the Python console. So once you have a Python console, you could able to visualize the tool. If you just remove this one, it is not going to be execute the program. Again from rdkit.com, import descriptors. Because you want to calculate the descriptors for the molecules, you have to call the package descriptors. Then all chemistry and data structure packages, and then import NumPy as NP. So all these are the importing has to be performed before running the actual task. So hope till here it is understandable. First you have to import RDKit, chemistry package, draw package, IPython console, descriptors, allchem and data structures. All these are related to RDKit and they are sub packages in the RDKit. Then you have import NumPy as NP. So because once you draw the structure, the descriptors are going to be in the number. Molecular weight is a number. Total polar surface area is a number. Then if you want to calculate Wiener index, it is a number. So without NumPy, it cannot give you the answer. So the NumPy will be for the handling the numericals. or addition, subtraction, any type of numericals can be handled by the numpy. Hence, we have to write it as a import numpy as np. So all these first 7 codes will be importing all the related packages. Now, actual task will be, you are going to define a molecule. You can give, instead of m, you can just say m1 molecule 1, and there is a particular format you have to follow, chem.mol from smiles, and you give the smiles notation of your molecule. The smiles notation is nothing but single line representation of the structures. So this can be anything you can give, either paracetamol smiles or any smiles you can give. Now you are going to execute the program. You can say run, or you can just click the F5 button in your keyboard. Now you can see there is no module named RDKit. That means the import has not been properly done. So now I can make one more cell over here. So sometimes because of online version, it is gives errors. But if you install already in your system, so for installation, you have to say pip install RDKit. Now you can see the star symbol says it is running, and I have to restart the kernel because I have installed a new package. So now if I run this program, sometimes online it gives you some errors. I have installed version in my system, but I want to show you in online itself because you have to do online. Every time whenever you are using new programs, you have to install all the RDKits. Sorry, online version is not working. I'll just open my already installed one. So once you have installed RDKit in your system, it is going to say that RDKit is going to be already installed in your system. Now we can run this program. So if you are having an error, it is going to give in the bottom line. If it is not error, so you won't get any output. Now we have imported all the packages related to the RDKit and ChemDraw, and we have written a statement molecule and convert this smiles notation, which is a single line representation notation, into the drawing structure. Now I just visualize this structure, just say mol, and you can see this is your paracetamol structure. So this you can copy paste into your system, save image as, or copy image, and you can put it anywhere if you want to save. Similarly, if you want to calculate descriptors for this molecule, initially we have just drawn the structure. Now we are going to calculate the total polar surface area. From rdkit.chemistry package, import the descriptors. This statement already we have given in the top itself. Either you can, before starting the programming, you can import all the packages once, or it can be individually, you can call the programs whenever needed. So in descriptors, you have a lot of descriptors are going to be present. One is total polar surface area, molecular weight, hydrogen bond donors, acceptors, Tanimoto coefficient, Wiener index, a lot of descriptors are going to be present. Now you can write a script stating that descriptors.total polar surface area, chem.mol for smiles, like molecule from the smiles already. Whatever the smiles you are writing over here, that will be converted into your graphical representation. From this representation, it is going to give you the total polar surface area. So here either you can give molecule 1 or you can give the smiles notation also. Just run the program and it is going to give you the total polar surface area of your molecule. So the important thing was how to represent or how to follow the path. And similarly, the notations. Like for example, if I just remove, I am going to just give some idea how the errors can happen. If I remove the closing symbol over here and run the program, and Python gives a error, unexpected EOF while passing. That means there was no closing statement was present in your Python script. So it shows you at which line the error is also happened. So at line 2 the error happened. So you have to go back and check carefully the line 2 and correct it. Now you can run it and you will get a results. It's not that there is no error in your smiles notation. There is no error in the calling of the packages. Only thing was how you have closed your program code. So similarly, we will see some of the properties. I want to calculate a log P, molecule log P. Descriptors is the same package we are going to call and calculate the log P of the given smiles notation. And at the end, you are going to type what is the log P of your molecule. Run the program, the log P will be 1.35. Without having any software in your hand, you can go to Jupyter Notebook and you can calculate the descriptors required for your study by giving the smiles notation. Similarly, so it's very important how you are going to represent the data in the Python. For that, you have to learn little bit of Python programming skills. For molecular refractivity, I'm going to again call the descriptor package dot molecule molar refractivity of the chemistry package. And yeah, just running the program. There will be error. I know that because I have not opened my statement. Chemistry.molecule from smiles, and you have to close the package properly. And you're going to get the molar refractivity 42.41. And similar fashion, let me explain you one more example. All these are not purely the machine learning. These are chemistry packages which are available. So these chemistry packages will help you to do the machine learning. Once you calculate all the descriptors, you can do the machine learning. So another task was to find out the similarity between the molecules. So whenever we are doing the QSAR studies or 2D QSAR studies, we want to find out the similarity between the molecules. Similarity between the molecules can be done by various calculations like fingerprint similarity, Tanimoto similarity, Wiener index similarity. There are 3000 descriptors are going to be present which can compare the molecules in different properties. So for that, I'm going to import data structures package. These data structures are very important, which have these fingerprints programs inside that. So from RDKit, I'm importing data structures. Again from rdkit.chem, fingerprints is a sub package. In that I'm calling fingerprint mols. That is the actual program or the command which calculates the fingerprint similarity. So here I'm going to define the molecules, and these molecules are going to be, I have to define the molecules in the Python by using rdkit, stating that chem.mol from smiles, and the first molecule, the smiles notation is 3 carbons and 1 oxygen, and the second molecule was 2 carbons connected with oxygen, and again third molecule which is also having 2 carbons in between oxygen. These are the 3 molecules I am defining. If you want to just visualize these molecular structures, you can just say molecules. So these are the 3 molecules which has been saved in the memory. Okay. Now I want to find out the fingerprint similarity between these molecules. So I have to return a statement or the program stating that FPS fingerprint similarity equals to, open your window, fingerprint mols dot fingerprint mol x for x in mol. So this is nothing but if you want to read out all the 3 molecules which you have defined in the package, should be read by using this particular statement for x in molecules. For each molecule, each x molecule in your list, that is molecules list, it is going to calculate and save it in the memory. So till here, by FPS fingerprints x in for x in mol, it is going to analyze all the molecules and try to calculate the fingerprint similarity between these 3 molecules and stores in the memory. But you want to visualize that similarity, you have to give a command data structures dot fingerprint similarity. So this is the command where you are going to visualize the what is the similarity between the 2 molecules. Then you have to write FPS 0. So 0 is nothing but the first molecule, and 1 is nothing but second molecule, 2 is nothing but third molecule. Whenever you are using a Python, as I said, the memory will be stored starting from the 0th position to the keep on going positions. So 0th position you have a first molecule, and the first position you have a second molecule, and the second molecule in a third molecule in a second position. So now you are going to see what is the similarity between the first molecule and second molecule. For that you have to write FPS 0th position at the first position. Just run the program and you can visualize what is the fingerprint similarity between these 2 molecules. That is nothing but 0.6 similarity. That means 60% similarity between the 2 molecules. Similarly, if you want to see the similarity between the first molecule and the second and third molecule, as I said, it starts positioning from the 0th position. It is going to be first molecule and the second position. So I'm going to see similarity between the second molecule and the third molecule in the list. You can see the similarity between the second and third molecule is 0.25. So you can say that these two are second and third molecules are same, 2 carbon atoms and 1 oxygen atom. But fingerprint similarity will be calculated based upon the positioning of the atoms in the structure. So when you see the positioning of the atoms, they are completely different and you are getting only 0.25% similarity. Similarly, I want to see similarity between the 0th position molecule, that is first molecule, and the third molecule, that is in the second position. You can see that is a 0.4% similarity. So like this, without use of any visualization tools and property calculation tools, by using RDKit, you can calculate the descriptors. So next, another important thing was how to visualize the charges which are present in the molecule. So if you want to visualize the gas tiger charges in the form of maps, maybe the chemistry people understand very easily. From rdkit.com chemdraw, you have to import the similarity maps between the molecules. As I said, you have to define the molecule in the particular format, chem.mol from smiles, and this is the smiles notation of your molecule. And you are going to compute property gas tiger charges. And these gas tiger charges can be computed by using various formulas. Like these two lines represent how the graph is going to be implemented. Let's see what is the output it is going to give you. So you can see not only the numericals, you can visualize what is the charge which is going to be located or present near the particular functional group. So these type of calculations, not only these descriptors and the maps, you have plenty of properties like 3000 parameters can be calculated by using RDKit. So hope all of you had a idea about RDKit, how it can be utilized. So let me once again explain you this simple machine learning model, as we already seen from sklearn. So now I am going to use sklearn for the programming. So I am calling from sklearn import tree, or you can directly say import sklearn also. And we have already seen this example. So now you see there is an error is coming at line 3 stating that because in line 3 labels, this apple has not been represented properly. You have to give it as a comma symbol, which was understandable by the Python. So if you learn Python, then only you could able to rectify the errors whichever it is showing. You can see it is going to predict 160 and 0 as a orange. So for example, I'm going to give 135, which is a weight of apple in between features like 130 to 140. But instead of smooth nature, instead of smooth nature of apple, which was defined as a 1, I am putting it as a 0, which is bumpy. So now you can see the 135 weight, which is corresponding to the apple weight, but the texture was bumpy instead of smooth. Now see how the program is going to predict this particular thing. It is going to give this as an apple. Why? Because, so if you see, truly speaking, it should be neither an apple nor a orange, because the weight is in between 130 and 140, which is corresponding to the apple. But the texture was bumpy, not a smooth. But still it is giving it as an apple. Why? Because the first criteria, what the decision tree is going to make was the weight of the fruit. The weight of the fruit is 135. So that is satisfying with the apple. Hence the output is coming as an apple. Why I am showing this one was, it's not compulsory that when you are giving a test data to the program, it may have mixture of different features. So the first query, whichever you are going to write, or the feature whichever you are defining, that will be checked first by the decision tree. Based upon that, it is going to give you the conclusions. Hope, is it clear for everyone? Hello.
There are few questions, Malika. If you are willing to take now, I will pose.
Just one more session is there, then I am going to have a queries.
Yes, then we can continue.
Yeah. So I just want to show you one more actual machine learning model. It may be bit high coding, like I can say the Python code was bit lengthy, but still I'll try to explain you. So the basic idea of this particular script was to understand you. As I said, when you want to do machine learning, you have to train the data, then test the data. So for training, you have to give the data. So this particular example, I am going to define 4 molecules, which I am calling it as a training data. I am not taking very huge data, only 4 molecules I am taking as a training data, specified in the format of notation which you have to define by using RDKit. M1 is equal to first smiles notation, second smiles notation, third and fourth molecules. So here you are going to use all RDKit packages and sklearn packages. As I said, there are 3 varieties of supervised learning packages are available. One is random forest, classifier, and decision tree. So in random forest classifier, we are doing the program, and all the molecules has been defined as a 4 molecules. Then fingerprints you are going to calculate. Once you define in the form of smiles, that has to be converted into numericals, that is fingerprints. Then again I have written convert the RDKit into NumPy arrays, where this particular for loop will be given. Once you learn the Python, you can understand how to define the for loops. For all the molecules which you have defined in the first training set, everything will be calculated, the fingerprints. So this is about how you are going to training your data by using random forest model. And in this, the ash symbol, wherever I kept and written few statements, generate 4 molecules. These are markdowns. They are not codes. They are not executable. Remaining things will be executable in this particular code. So once you write this one, you run the program so that your training of your model will be done. So once the training is over, the next thing is you have to test your model. So once the data has been generated by using 4 molecules, you are going to write a simple code. So it is going to generate a best fit model. So always you have a x-axis and y-axis. You are trying to draw a best fit line between the x-axis and y-axis. So in this particular model, I have 4 molecules. In the 4 molecules, 2 molecules I made, first 2 molecules I made it as active molecules, and the next 2 molecules as inactive molecules. So the same statement I have written here, the first 2 molecules being actives, which will be represented as 1, and inactive molecules are as zeros. So this rf.fit, That means random forest dot fit. Whereas in case of first model in apple oranges, we have written classifier dot fit. So the classifier is going to make a best fit model. But in case of this particular example, random forest model is going to make a best fit model between the y-axis data, actives and inactives. So till here, it is going to be the job of your model to get a training of your model. So once the training is over, this is the new molecule, that is the fifth molecule M5 with a different smiles notation I have given to predict whether this molecule is going to be active or inactive based upon the fingerprints. So for this particular new molecule also, the fingerprints will be calculated by using the formula same code, data structures, and all the fingerprints will be calculated and print whether this is going to be a active or inactive. So when I run this particular program, you are going to get an answer as 1. 1 is nothing but active molecule. So this new molecule based upon the fingerprints which was taken from the training data, it will be predicted as a active molecule. This is how the simple machine learning models can be written. This is simple model with only 4 molecules as a training set and random forest model. But when you have a huge data with different parameters, like I have a molecules which are having log P and molecular weight and hydrogen bond donors, these 3 are going to affect the IC50 of your molecule. So during that case, you have to use the multiple linear regression model. So first of all, you should know the data nature. Based upon that, you are going to have a output. Hope this will explain you like how this is going to be predict the molecule nature. So is there any queries related to this session?
Good afternoon, ma'am.
Yeah, good afternoon.
Can I just ask the questions from previous session also, ma'am, if that's okay?
Yes. Yeah, definitely.
Pooja, first complete questions from this session, then we can go back to the first session.
Okay, ma'am. Okay. So from this session, there is one question from Mr. C.R. Patel, who asked how the arrow has come.
Which arrow?
You were typing the programming, ma'am, during which there was this question. So you were working in the Jupyter notebook, and during which they were trying to ask you how did the arrow mark come from?
Yeah. Okay. It's very simple. Let me share my screen. So when you just click on enter button, so let's see, this is the last line where I'm writing hello world. Okay, then say run. The next cell itself comes with an arrow. If not, you have to just add this insert cell below option. Then the next cell is going to be added with arrow. So this arrow is basically the code. If you want to make it as a markdown in this particular drop down window where I've just clicked my cursor, if you make it as a markdown, the arrow goes. And if it is a code, the arrow comes automatically.
Next question is from Mr. Keshav. Is it a sort of programming?
Yeah, it's a sort of programming, definitely. Python is a programming. Once you know the programming, then only you could able to do the machine learning.
Next question is from Dr. Mayank. Madam, is there a user manual for commands to be used in Python software?
Yeah, lot of online commands are available. But thing is, whichever is important for your work, you have to segregate the data. Because reporting data which is related to tables is different and images is different. There are a lot of online Python courses are available. You can check it out.
Next question is from Dr. Komal. Is it applicable for only those structures which are available in a particular package?
No. There is no availability of the molecules in the RD kit. Whatever the smiles notation you will give, accordingly it will draw the structure.
Next question is from Prashant. Ma'am, how to give SMILES notation for large molecules like proteins or polymers, etc.?
Proteins and polymers cannot be given in the SMILES notation. They can be directly imported in the form of .pdb format or .sdf format. When you have a ChemDraw with you, you draw the structures and you save it as a SDF format, you can import. If you have protein structures from the PDB data bank, you can download and you can directly import into the Jupyter notebook.
Ma'am, this question is from my end, ma'am.
Okay.
Is it possible to import large PDB files like protein structures, example, in Python, and you can actually do the molecular docking studies in here?
Yes, yeah. But the script is different because during that case, you have to import even Autodock tools or the Schrodinger tools, whichever the software where you want to do the docking studies. That particular environment of the Python has to be imported into the Jupyter notebook. For example, if you are working with Autodock, when you go to the Autodock website, they have Python scripts already available. You have to import that Python script first, then you import your PDB file.
So, ma'am, whatever we are working down, like in Autodock or Schrodinger, those tools can be imported and entire thing can be done in Python itself.
Yes.
Okay. Only thing is like little bit of coding knowledge.
Yes, yeah.
Okay, thank you. So that's it from this session, ma'am. There are questions from the previous session, like morning one.
Yeah.
So the first question is, Madam, can you explain about ADMET studies?
Yeah, ADMET is nothing but absorption, distribution, metabolism, excretion, and toxicity. So all these properties are very important when the molecule will be taken orally or any other route, how it is going to be absorbed, what is the absorption rate, from where you can have a area under the curve and T of values which you calculate from the pharmacokinetic studies. Similarly, the toxicity like mutagenicity, carcinogenicity, and some of the genome toxicity. So all type of toxicities can be predicted, but they are not very 100% accurate, but you will have 90% of idea about how this molecule is going to be affecting on various DNA structures or in the genomes which are present in the body. Similarly, the elevation rate, excretion rate will also help you to what is the half-life and how much time it is going to be present in the body, and metabolism, how it is going to be affected by the CYP enzymes. So this data is good enough to understand what is the nature of your molecule in the body.
Next question is from Dr. Arifa Begum. Is this body on chip directly used on human volunteers, madam?
It's not going to be used on any human beings. It's just a chip, microchip, which is going to be present in a it's like a slide, you can say, which is having a top and bottom slide. In between, the connectors will be there with respect to the all the different cells. It is not going to be implemented or injected or kept it on any of the bodies. It's a simple in vitro chip where you are going to inject, but it mimics the human environment by the conductors which are going to be connected with the various organs.
Next question is from Sindhu, ma'am. Madam, can we avoid animal study for activity by using artificial intelligence?
I cannot say 100% you can avoid, but the number of animals used for the study will be reduced definitely.
Another question from Sujit, Mr. Sujit. Is QSAR study necessary before going forward to AI and machine learning drug discovery?
There is no hard and fast rule that you have to do QSAR before doing AI and ML. AI and ML itself is a one type of statistical study, because whatever the statistical models we use in the QSAR, similar type of QSAR like simple linear regression, multiple linear regression, all we are going to use in AI model.
Other question from Dr. Arifa Begum. What in silico tools are useful for detecting mutation level predictions?
Okay, there are plenty of online tools are available. One is iPlant, then pathogenic toxicity predictors. iMutant can give you the mutation severity that you can try. like stability of the mutated structure tells you how much that particular protein is capable of that mutation. If the stability is very high, you can say that that mutation is not going to be change the structure. But if the mutation stability was very less, the score whichever you are getting is very less, that says that that is going to impact your structure very high. You can try I mutant.
So, another question from same participant, ma'am. Can you please tell the importance or role of our students in this machine learning?
Yeah, it's not only pharma students. Everybody, if they learn machine learning, next another boom will be all companies are diverting towards the machine learning. And there was one great quote stating that if you're not going to learn machine learning, you're becoming a lion. Like that, some statement was there with some great scientist. Like because most of the jobs will be taken by the machine learning scientist in coming years. So it's better to have if the person is having scientific knowledge along with the programming knowledge, then it will be added advantage for the company as well as for the students also.
Ma'am, then in that case, will there be replacement for human also by the computers? Because if everything is auto-coded.
No, it's not like that, because computers also you have to train. But for training, you require programming skills. So there is no 100% replacement of humans with computers, but there will be reduction in the number of intakes.
Okay, ma'am. Thank you.
Participants, do we have any other questions? Please post in the chat box. Okay. Sridhar, madam, shall I conclude the session?
Okay, Rama. If there were no questions, then you can conclude.
Okay, thank you, madam. On behalf of the participants and behalf of Institute of Pharmaceutical Technology, I extend my heartfelt thanks to Dr. Mallika for her elaborate and continuous presentation for 3 hours. I really appreciate your stamina. You are able to lecture continuously for 3 hours. The examples which you have quoted, I'm always attracted towards the simple examples which you always quote. Whether there's an apple, banana, or simple, you made us understand what is the difference between supervised and unsupervised learning. So we really thank you for your presentation and even making us understand how to utilize the programming in the drug discovery field. Thank you once again, Mallika, for your time, patience, and your valuable information given for continuously for 3 years. Thank you.
Thank you, ma'am. Thank you all the organizers for giving this opportunity. Thank you one and all.
Thank you, Mallika. Thank you for accepting our invitation. We also expect same cooperation in future also.
Yes, ma'am, definitely.
Thank you.
Yeah.
Thank you very much.
We expect you physically next time in our department.
Yes.
Yes, definitely. Thank you, ma'am.
Thank you.
Thank you.
Ma'am, next afternoon session at what time we will have?
Session is at 3 o'clock, by
Okay.
Bharatam sir. Okay, you
I
just
request all
that.
the participants to join back at 2:50 for afternoon session by Professor Bharatam Garu. Sir is a bioinformatics specialist from Naipur, Mohali. So I request all of you to join by 2:50 itself. Thank you.
And also one more announcement, mock test.
Today evening by 6 o'clock, a link will be provided to all the participants for mock test. The guidelines for mock test has already been circulated in your WhatsApp group. The link will be active only for 10 minutes. It's only for your convenience. We request all the participants to attend the mock test so that they will not face any problem in tomorrow's test. Thank you.
Thank you, Sujata. Thank you very much.
Madam.
Thank you. Okay.
Bye, madam.
Tomorrow afternoon we will meet.
Meet at 2:50. Okay, bye.